all the students and all the respected faculty members, my dear colleagues, for attending this talk and giving me the opportunity. So I have designed my talk more sort of like a popular talk, where the basic theme will obviously be proteins, which is like my central point of research till date, and we'll try to extrapolate the dynamical system behavior, or how proteins interact among themselves, and mostly based on protein evolution in a macroscopic perspective, and we'll try to draw some analogies, empirical observations, supported by empirical observations and certain semi-empirical theories to the domain of social science. And we can brief it like this, that what are the similarities and differences, if at all, between the protein society and, let's say, the human society. So to go along with it, to start, I mean, this is a heterogeneous batch, although you are, all of you are from my department only. So I believe all of you know the basics about proteins, but it's never a bad thing to repeat something important. So proteins are really important, and to a layman, to a passerby in the road, if you casually ask what are proteins, will probably redirect you to some some supplement that we used to have. There are a lot of protein supplement these days. So basically, in a layman way, we understand that that's a very essential food component, and we often, relate the average health of an individual, whether he is slim or he is really slim, so you require more proteins. So that way, that's like a layman way to think about it. But since you are a bit more than that, so you all of you know that protein is the functional biomolecule, so the functional unit of life in a molecular sense. So so macromolecule, all of us know that life consists of several macromolecules, and they are essentially polymers, and most of them are heteropolymers, except for a few carbohydrates. So all of these lipids and carbohydrates, as well as proteins and other hybrid compounds also, they have this polymeric nature, and that's very important for the persuasion of life. Among these macromolecules, the first macromolecule that was known to have some functional activity was ribonucleic acid. As we all know that the primitive world was an RNA world, and RNA was a self-sufficient, self-competent molecule. It could by virtue of having a nucleotide sequence, it could be replicated to another strand, and it could also do some catalytic stuffs. So like ribosome, as we know, in ribosome we have RNA and protein together, and after that, from RNA there was certain realization of nature that RNA might be very susceptible to, let's say, alkaline catalysis, alkaline hydrolysis, for having cis-diol, etc. So it and other factors also prompted nature to get a double-stranded molecule, that was DNA. But DNA never really had a catalytic activity, so they required another third molecule. So one is for safekeeping. There is one in between which plays several roles, like messenger RNA, as we know, and there are tRNAs which are like transferring molecules, and there are these ribosomal RNAs and others as well. And then finally, we want to execute, we want to make certain functions always going on, and that's 97 to 99% of biocatalysis are actually performed by proteins as enzymes. So if you are asked that what is the classic type function in proteins, you always answer that is enzyme catalysis. but there are, this is a nomenclature that has lately come up, like dividing protein functions in terms of classic and non-classic type. But so that's not really restricted to the classic type function, like enzyme catalysis. And as we all know, let's recall that enzymes are catalysts, biocatalysts, which remain unaltered before and after the reaction. So it never really changes the direction of a reaction, things like that. For that, there are other mechanisms. For example, enzyme coupling is one mechanism, and high energy involvement of high energy compounds, that's one mechanism. So it can never really transfer a forbidden reaction to an allowed reaction on its own. What it can do, it can decrease the energy requirement by virtue of creating a transition state. But before and after, it will remain as it is. So that's enzymes. So among the non-classic type functions, there are protein-protein interaction. Protein-protein interaction plays a very key role for several purposes, and one such purpose is signal transduction, signaling molecule. We were studying in other virology classes, etc. I recently taught that there are some antenna molecules of a cell, which we call receptor, and which are membrane proteins, essentially integral membrane proteins, and they are always protruding out from the cell, and they will receive certain signals, and they have some three domains. One is outside, so that is like the extracellular domain, then one is in between the membrane, embedded in between the membrane, so there is a transmembrane domain, and then we have the cytosolic domain. So whatever happens at the outside will have an influence, it will have a long-term effect at the tail. So that's like the cytosolic tail, and then other molecules will be recruited. There will be a cascade of reaction eventually leading to transcription of certain genes. So that is signal transduction for you. And then several other function proteins participate, like in anchoring as a scaffold. Membrane proteins are classic examples again. Providing structural framework, so collagen, keratin, all these fibrous protein, and also in locomotion, in movements, in working as a motor protein. You have several kinesin, dynein, all these examples are there. So proteins are very, very important. So that's why protein. We have to, if you are studying biology, we cannot do without protein, as simple as that. And these days, after gene therapy, there's something, another technology is coming up, leaps and bounds, and some in a few years time, maybe in 10 years time, it will be at your doorstep. That is called protein therapy. So if you just like an extrapolation of gene therapy, so obviously things will be manipulated at the protein level, and it will be directly used as a medicine. Protein design is another area where people are working, and it has many applications. So proteins are important. Antibodies are proteins, the other important thing. And this is again a basic, all of you know, I'll just read through. So it is a heteropolymer of 20 naturally occurring amino acids. So they have hydrophilic components, polar components, and hydrophobic components. So polar, hydrophilic are the same roughly. And amongst the hydrophilic component, we have charged and uncharged. So uncharged are basically polar residues. So for the charged residues, we have lysine, arginine, glutamate, and aspartate, and positively charged histidine, because that's a physiological buffer. And then the other, asparagine, glutamine, which does not have a charge at your physiological pH. So those are the polar residues, and then again you have hydrophobic residues, and then we all know it looks like a beads on a string. So if you can imagine, you can give a simple analogy of a it's like a necklace, or some beads on string stuff. We have different colored beads on that. So if you have 20 colors, so there are 20 amino acids. So it's like that, and then they are foldable globules in polar solvents. So that means because of having these hydrophobic amino acids, which cannot sustain the presence of water, they will always look for an event which is called hydrophobic collapse, and it's an entropically driven event. So that's a very fast event. So whenever you can do an experiment like dropping oil drop in water. If you take a container and pour water in it, and then drop a oil and then drop another oil from another end and do a little shaking. So you'll eventually see that the two drops will coalesce into a single one, because it maximizes the entropy, and likewise it minimizes the free energy of the system, of the overall system. So that's a classic example of a hydrophobic collapse. So what happens, because oil cannot sustain the presence of water, although that's amphiphilic, so there are hydrophobic components of the oil. Oils are essentially lipid, so they cannot sustain the presence of water, so they will try to find each other, and they come under the same umbrella because they have a common enemy in water. Until date, people haven't been able to find any atomic force for hydrophobic effect. So that it's not that they have attractive forces among themselves, the hydrophobic residues that I'm talking about, like hydrocarbon. They do not have any polar component. But since they have a common enemy in water, they come in close contact under the same umbrella, and that's called the hydrophobic effect. So when these, in case of a globular protein or any protein which is ordered, it could also be fibrous protein for that matter. But it's an ordered, it has to have an ordered structure. So what happens, all these hydrophobic residues will try to find each other, and the process will be very fast, because it's very unfavorable for the whole system to sustain exposed hydrophobic residues. Exposed means exposed to the solvent, water, because there is no interaction, there is repulsion actually. If you take two bar magnets and if you place the north poles, side by side, you'll feel a repulsion, so that the same repulsion is also happening here. So they will try to find each other very quickly, and the process is very fast. And then other residues, the polar and the charged residues, they will accordingly align, and the overall shape will be that of a globule. Globule means roughly spherical. And as we all know, if you can imagine all the three-dimensional regular geometries that you can think of, like a parallelepiped or a cone or a cylinder, sphere is the most symmetric one, isn't it? Sphere is the most symmetric one. For that, the planets are spherical. All the units from microscopic to macroscopic level, we find these spherical objects because it has the most symmetric geometry respect to a cube, parallelepiped, and other regular three-dimensional geometry. So they will try to find each other, and the overall shape will be spherical or globular. Sometimes elongated proteins as well, but mostly, they will also be part of the globular protein category. But most proteins are roughly spherical. They try to mimic a sphere because of that symmetry. And so the other thing, have you heard about Levinthal's paradox? Any one of you? Levinthal's paradox? No. Levinthal's paradox, in very few words, was a hypothesis. It's not is, it was a hypothesis and a paradox which enumerated the folding time of a 100 amino acid globular protein by systematic sampling. In systematic sampling, proteins, when two amino acids, that they form the peptide bond, of course. So that's a peptide bond here in C. This is a partial double bond character in trans conformation, except for cis proline. So there are conformational sampling in a few words, and Levinthal's tried to enumerate the folding time for a small size or medium size protein, let's say 100 amino acid long. If the conformational sampling were systematic, like every conformation is systematically sampled at a rate which is the rate that we encounter within the cell, and that's very fast, believe me, 14 per second. So in 1 second you have 10 to the 14 conformer samples. Even with that high rate, it was mathematically computed that the folding time required for a 100 amino acid long protein to find its folded structure, which is its global minima structure, is more than the age of the universe. So obviously there was something wrong, because proteins fold in second to millisecond in that time scale. So proteins fold in second to millisecond in that time scale. Sometimes it goes, like a minute or so, but never more than that. So there was obviously something wrong. with it. But this paradox existed in the literature for more than 40 years before it was eventually explained by Trifonov, who in 2000, in the decade of 2000, he proposed this model of co-translational folding. In a few words, it is like proteins are heteropolymers, so it has to satisfy certain physical constraints. It cannot avoid the heteropolymer physics, and one of such physical attributes is that a heteropolymer can propagate in a linear direction only up to a certain length, and then it has to take a turn. That was his proposition. It cannot go infinitely in a linear direction in space. So that turn, when encountered, when considered in the proposition and put in the Levinthal's equation, like we have some 20 to 30 amino acid long units. So if you go to the protein data bank and find out all the helical units or a beta sheet unit, the average length will be 25 to 30 amino acid long. In space, of course, I'm talking about here. So when that was considered in the Levinthal's equation, it actually hit the millisecond to second time scale. So that was the approximation. That was the oversimplification in terms of Levinthal. So the paradox was no more, and everyone realized that it is a co-translational folding that happened. So what happens during the process of ribosomal protein synthesis, or translation as we commonly call it, the protein is being synthesized, the growing polypeptide chain is being excised through that E site of the ribosome, and it just does not remain in the solution like a random coil like that. Once it is exceeded, gradually, then on, concomitantly, it takes the shape. Whatever local interaction is possible, they start to happen. It's not that the whole protein is first synthesized and thrown into the cytosol as a random coil and then folding occurs. Never like that. So it is a co-translational folding. Folding is simultaneously happening with translation. So helices, that's why, are more stable secondary structure than beta sheets, because helices use intramolecular hydrogen bonding, while beta sheets use intermolecular hydrogen bonding, or interchain, interstrand, whatever you call. So, a smaller patch standalone has a greater chance of hitting a helical secondary structure rather than a beta sheet secondary structure. I hope I make sense. Then the structural, so Anfinsen, you have to remember his name if you are into proteins or anything related. So Anfinsen was the first in 1960s who proposed this sequence to structure conservation. So he took RNAs as a model system and then put some denaturant into it gradually, and saw it got denatured, and then he removed the denaturants from the solution, and then again the protein got renatured. But he did his experiments only to RNAs, which are like 50 to 100 amino acid long. They're small proteins. But his conclusion was that all the information required for the three-dimensional structure, or the knowledge of the three-dimensional structure, how it would actually take shape, how it would look like, after as a folded entity, the information is already there in the primary sequence. So that was his hypothesis, because of this reversibility, in denaturation and renaturation. So you include urea or guanidinium hydrochloride, you let it denature, so it will be eventually unfolded to a random coil, and then again you wash out all the denaturant, you will eventually find the folded protein back. So it's at his time that was a big a path-breaking proposition, that all the information are actually embedded within the primary sequence only. There is nothing fed externally. But that was the time of 1960s. After that, obviously, the protein data bank explored. Nowadays, you will find some 100,000 structures in the protein data bank. And the first crystal structure that was solved for proteins was that of myoglobin, and that was made publicly available in 1950. After Ramachandran plot, so it was in 1960s only, although it was solved in 1958. So you can imagine the difference of the time scale of these experiments. So nowadays, obviously this does not hold true. So that means many sequences can actually have similar structures, or which we technically call a protein fold. So many sequences, even if they have less than 20% sequence identity, even if they have less than 10% sequence identity, they can still map to the same fold. So this is called the fold prediction problem in proteins. Fold prediction problems in proteins. It's a tough problem, although this is kind of solved. And I also in my PhD days worked on this problem. Many others did as well. So this problem is not really relevant in today's time. It is kind of a solved problem. But we know that there exist what they call twilight zone and midnight zone of protein folding, which means you have two sequences having roughly the same structure, that is, they are hitting the same fold. Fold has a technical definition, I am not really going into that. They are hitting the same fold, although they have less than 10% sequence identity. They are not really similar in terms of sequence. So then they fall under the twilight zone. And if that number comes even less, under 5%, less than 5%, and again they hit the same fold, that is called the midnight zone of protein folding. So, so obviously it was realized that structure is more conserved than sequences. And why the structure is more conserved? Because structure is directly related to function. Because it's a three-dimensional thing, and function is always happening at the three-dimensional level. So, function governs the evolutionary principles, and that determines the structures. And structures, then evolution evolution is never stopping. We are still evolving, right? It does not have a stop. So what evolution tries to do, what nature tries to do, to preserve certain functions intact and then play around to explore or check if there could be more functions. And as we are growing, there is almost limitless growth, which is kind of absurd. But consequences will be faced. But anyway, as we are growing, we are becoming multitasking day by day, isn't it? Imagine our fathers and forefathers, what they used to do, and imagine what we are doing. We have to be skilled in a multitasking manner, and that is also happening at the protein level. That is also happening at the cellular level. And this is not really drawing a conclusion empirically. This has been observed that with more time, people have found more multifunctional proteins, where nature has preserved a function, a native function or a mother function, parental function, and then additional functions were evolved in the same protein, but in a subtle manner. I will go into that in one of the next slides. So sequence, structure, function, we can also say one sequence, one structure, one function. That was the paradigm. Nowadays, and here comes the emergence of intrinsic disorder that has completely violated that paradigm, and people have seen that first of all, many sequences can lead to a same structure, which can lead to a same function, and since evolution is ongoing, there is always a demand of more functions, dil mange more, and that's also true in protein level as well. So multifunctional proteins are evolving, and one of such mechanisms that nature has figured out is to create something called intrinsic disorder. So what is intrinsic disorder in a simple way? I'll come to that, but before that we have globular protein, membrane protein, fibrous protein. We can consider all of them in a same group, which are ordered. And whatever left aside is intrinsically disordered proteins, and they also there could be proteins which are completely disordered, there could be proteins which are partially disordered, there could be disordered peptides as well. So all these constitute another class which we call disorder in brief. So as you see, disordered to structured, there are different synonyms, structured, unstructured, ordered, disordered. These are the similarly used terms, synonymously used terms. So why intrinsic disorder? So what is the definition of intrinsically disordered proteins? Those proteins which never finds a stable structure. That is a simple definition. They never find a particular fold. They can be this, they can be that, they can take many different conformations. And why is that? To answer that, we have to go to a future slide first, I think. Yeah, this one, and I'll come back to that. So this is happening because of the energy landscape. So in case of a globular protein, if you draw the free energy landscape, there is a deep well, there is a deep sharp well, and once you are close to that well, eventually you will fall into the well and you will be stabilized. So that is the well here, as you can see. It's a deep and sharp well, but there are obviously some crevices on the side, as you can see. It's not really a perfect regular geometrical shape, of course. So there are local minimas where you might get entrapped for a while. So those proteins can lead to molten globule-like structures. They are called molten globule. They have secondary structure, but they completely lose their three-dimensional or the folding attribute. So secondary structure is again local interaction, but how helices and beta sheets interact together to form the fold is tertiary interaction. So molten globule does not have any tertiary interaction. The other important thing in contrast to globular protein, for intrinsically disordered protein, there is not a single global minima. There are many global minimas, and they are kind of similar in terms of energy. So the energy landscape is rough and it is rugged. It's not like a well. So that's the basic reason, and that's why intrinsically disordered proteins belongs to the restless category. They never find a stable solution. You can draw analogies to, let's say, the in terms of atoms, or in the periodic table. You can look around the atoms which are closest to the inert element, the halogens, for example. So they will they just need one partner. So they have low valency compared to carbon, let's say. So carbon needs 4 of them to get that outermost electronic orbital conformation like a nearest inert element. So it's like that. So intrinsically disordered proteins always look for partners so that they could be aligned to them and get one of these stable conformations back, but they never find, and they just hover around different local minima in the energy landscape. Going back to where we left here. So these are the common functions of proteins. You can see they can act as growth hormone, for example. Insulin is a protein hormone. There are tyrosine kinases as receptors. They are also proteins. So so they can also act as receptor molecules. They can act as transport molecules, like locomotion. Structural molecule, actin, myosin, all these ones. The contractile molecules, enzymes, of course, and antibodies. So all these are regular functions, and mostly they are performed by some or the other ordered protein, globular protein, let's say. So they kind of attempt to perform certain routine functions, all these ordered proteins, routine functions. So a particular structure mapping to a particular function. So in contrast, if you look at the roles of IDPs, there are actually more. Ordered assembly of ribosome and chromatin, then recognition, binding, transportation through nuclear pore, RNA chaperoning, protein functions, nucleic acid binding, flexible entropic linker, regulator of signal pathways. So there are many functions which IDPs perform, actually more than what ordered proteins do. So ordered proteins, by here I mean globular, fibrous, as well as membrane proteins. So they all have ordered structures. So, but the thing is, IDPs, although they can perform multiple roles, they are not stably sustainable with a routine life or without a binding partner or on its own. it will always look for it's restless because it is trying to find a global minima in its energy landscape. There is none, there is only some local minima. So in search of that, it is always hovering, always, never stop searching. While a globular protein, once it is close to that well, and we all know that if you are at the valley, then obviously you are very stable. But if you are at the peak, you are very unstable. A little trigger, a little toka can actually bring you down to that valley. So once you are at the valley, then you are stable because you require huge amount of energy to pick you up again to the peak, right? So once they are within that global minima well, so they are stable on its own and it knows how to perform a routine function, and does not really require another fold or anything like that. So that it's kind of routine. But for intrinsically disordered proteins, they are restless and therefore they are always looking for partners, and they're always trying to do some or the other work, and several functions are known where they perform, or participate actively. So structural order is meant to perform routine or prototypic function. There are many examples. So I'm giving one example for each type. For enzymes, let's say this is glucokinase or hexokinase performs the same role. If you look at the structure, it has a dimer. This is a dimeric structure, and then there is some allosteric regulation here also involved. It has an ATP binding pocket, it has a glucose binding pocket, and the glucose binding pocket, the ATP binding pocket is filled up with water in absence of ATP, so it requires the ATP to kick out the water molecules, and then only this reaction can take place. The reaction is a nucleophilic attack, and so on and so forth. So this has existed in nature and existed in nature throughout evolution stably. Hasn't been a lot of difference between a human glucokinase or let's say a rat or a mouse, or even in a lower species, wherever you find glycolysis is kind of common. So if you take all the hexokinases together, do a sequence profiling or sequence alignment, you'll find very high sequence similarity as well as structural similarity because the function has been conserved. And they're the same for many other, like anchoring and motoring. This is like the motor proteins that we are talking about. It's just I'm showing some prototypic function and routine functions, and then we are going to the membrane proteins. So obviously, this is not a membrane protein, of course. This is a two globular protein interacting, and it could be mainly it is for signal transduction, somewhere involved in signal transduction, but it could. So signal transduction essentially means it will eventually lead to the transcription of some gene. So if you discard that last point, there are certain reactions otherwise also happening, which does not really lead to the transcription of a gene, but there is also a cascade. So protein-protein interaction is common and common for all. And then you have membrane proteins. These membrane proteins receive signals, transduce them. You have channels. I have not really shown aquaporins, porins, or the channeling. So all these functions are performed by some or the other ordered protein, and which are like that and conserved through evolution like that. So here you can say that one function, one structure, not really one sequence, but closely similar sequence. That sort of a paradigm is followed. It does not really violate too much. This is collagen on your left, and this is keratin on your right. So it leads to the hair and muscle, and these are structural protein along with actin, which are required for cytoskeleton, etc. So these proteins are also there. Again, this is fibrous protein, again ordered. But then there has been instances of emerging novel functionalities in proteins with time. One such example, and I'm very familiar with it because this was one protein where my PhD lab had a lot of interest, and they solved the crystal structure, etc., etc. So I know this, from very direct experience. So this is cyclophilin. Cyclophilin is primarily a cis transpeptidyl prolyl isomerase. So it takes the cis transproline and then do that isomerization to convert into a cis proline. And it is a beta barrel. As you can see, it's a barrel-like structure. It's like that. It's like that. It's a beta barrel, and it has a breathing motion. So which is like a normal mode. It's always like breathes like our lungs, you know. And so after when people started exploring the functionality of cyclophilin, they quickly recognized that this is this can be a very good drug target, a potential drug target for organisms like Leishmania donovani. Leishmania donovani has cyclophilin, and that's an essential protein for their purpose. So if we are able to inhibit or kill or inactivate that protein, we'll eventually kill the organism. But at the same time, we'll have to keep the host safe, isn't it? So that's called selective toxicity. So when researchers went through that trajectory, they found out that upon binding to this drug called cyclosporine, which is a cyclic peptide, and it has non-naturally occurring amino acid, and it's a immunosuppression. Whenever you are doing some organ transplantation, so you use cyclosporine, but then your immunity becomes off and you are vulnerable to infection, so you have to keep safe from other infection, etc. So cyclosporine is this compound, as has been shown here, is the crystal structure, and this was actually solved in our PhD, from where I did my PhD, that laboratory. So they found out that unfortunately this cannot be used as a drug target. Why? Not because that this is this cyclosporine is also binding to the human cyclophilin. So we are targeting Leishmania donovani cyclophilin here. The threat is that, the direct threat is that the drug may also bind to the human cyclophilin, but that was not the thing which was happening. They have some 70% similarity in their sequences, but human cyclophilin does not really strongly bind to cyclosporine. But the reason was this, that upon binding to cyclosporine, that complex is now recruiting another third protein, which is a human protein, which is called calcineurin, which is shown here. And this is so this is your CNA. So this is your cyclophilin, and this is the entire thing. Calcineurin, again a heterodimer, it has two different chains, etc. And that calcineurin is essential for human purpose, for certain metabolic pathway which cannot be compromised. So then that ruled out the use of cyclosporine as a drug against leishmaniasis. So here my point is, here that upon evolution, and the same protein is found in across species with time, etc. So upon evolution, subtle changes has happened, even involving intermediate binder, intermediate compounds. And in some of you may know that there are water mediated interaction sometime happening in DNA protein, etc. Especially in DNA protein interaction, which we call indirect readout. So water mediated interaction. So let's say this is a protein, then this is a DNA. They are not binding directly, but through some water, through forming hydrogen bonds with water. That's called indirect readout. So it's the same thing happening here also. Upon binding to cyclosporine, the entire complex becomes susceptible very heavily. So binding affinity was very high with calcineurin, and that's something that has evolved with time in this protein, in this class of protein. So these changes, these functionalities have been evolving. Commonly this is actually called protein moonlighting, or it's also called gene sharing. So the idea is that you conserve a novel function, a parent function or a mother function, native function, whatever you call, and then do the tinkering at your non-active site, mostly at the surfaces, because proteins do want to keep the hydrophobic core intact, but there has been instances where there has been mutations in the hydrophobic cores as well. We also work on that. that alternative arrangements of packing can also lead to the same hydrophobic core. And the other residues, they undergo certain mutations, and sometimes the mutations are deselected evolutionary if they are not fit to survive, like following Darwinism. And some, if they are fit to survive, then they might lead to some other function, but not at the cost of compromising the original function. So that's the whole thing. And the other very simple idea here is that to split the functions up into domains. So that is like another parallel. So parallelly evolved multi-domain proteins. So as you can see here, 3 distinct domains. Domain, by the way, is a self-foldable unit. So if you just use a scissor, a molecular scissor, to cut the peptide bond between those domains, and then try to fold each domain independently from its sequence, you will find that the protein is able, the domain is able to fold. Like in virology class, we were discussing receptor binding domain, spike, same 3 if anyone around, spike receptor binding domain. So that's an independently foldable unit. So while assigning multiple tasks, while assigning multiple functions, nature has used, so it has to do it in the genetic level. So it has used duplication and then gene sharing, and duplication is very important actually. So if you take a copy of the of one gene, then you can keep the primary copy unchanged, and then do some tinkering with the Xerox copy or whatever, and see if that works. So that is another way nature has worked to explore multiple functions of proteins. There has been emergence of fold switch proteins, if you are aware of it. So there are proteins which switches its fold. So you can see here, and this is when there is a certain environmental stress, let's say a change in pH or temperature, then certain protein switches their fold and obviously perform certain other functions. And of course there are these intrinsically disordered proteins, which is like real breakthrough in terms of evolution, in terms of functional evolution of proteins. So where intrinsically disordered proteins is also called highly dynamic biological soft matter, because it has liquidity in contrast to globular proteins, which resembles crystalline solids. These are, as material, they are more liquid. So that's why they can align onto different surfaces and take the align accordingly onto different surfaces and take different conformations, just like water. You pour it into a bottle, it will take the shape of a bottle. You pour it in a glass, it will take the shape of a glass, and like that. It's not really purely liquid, but it's highly dynamic biological soft matter. So one key mechanism to achieve that is to use multiple seeding points. If you have heard of nucleation condensation, so an event starts from one point and then it spreads. So that's called nucleation condensation. So in case of intrinsically disordered protein, there are different seeding points upon binding to different partners. You may ask that how it has the flexibility to bind to anything and everything. It doesn't really bind to anything and everything. It also has there is an order of disorder also. It chooses, of course, more than one, but not anything and everything. But it has certainly, this has been found out that it has different seeding points for different interactors. This is one of our work. I just thought of incorporating this slide. So one key mechanism to retain the flexibility in proteins, in intrinsically disordered proteins, is salt bridge dynamics. So salt bridge means there is an anion, there is a cation, like you have a arginine or a glutamate, they come close, and there is a electrostatic interaction between the charges. So that is a ionic bond or a salt bridge. And we have seen, and other people have also seen, that the salt bridges interchange, and intrinsically disordered proteins have an abundance of charge residues, but not really biased towards positive or negative charge, not like histone. They have abundance of charge residues. So a obvious question is that how they can retain, because how they can retain the desired flexibility even in spite of incorporating so many charges, because charges will lead to salt bridge, and salt bridges will bring local rigidity, isn't it? It will bring together distant parts of the chain, and there would be a formation of loops and turns and things like that. So how is it possible that both these seemingly contrasting phenomenon can coexist? So we had figured out how is this possible, and very well, it has some 20 citations also nowadays, a 2017 paper. But the basic idea is that that there are ephemeral salt bridges. There are salt bridges that forms and collapses, and always they are interchanging partners. For example, in this slide, this is A beta 42, or the causative protein for Alzheimer's. The amyloid beta has 42 amino acids. There is one lysine, that's the only lysine in this protein. May not be that clear, but trust me, that it was found to interact with several other negative residues, and these are different conformations picked up from the molecular dynamic simulation trajectories and superposed. So it was always found to interchange their partners. It was never stable. So that's how different parts of the polypeptide, and imagine this happening throughout the chain. So different parts of the polypeptide chain are coming close at different instances. Therefore, different conformations are being sampled. So that's how they retain their flexibility. We also extended this to COVID research and found its functional implication in spike furin interaction. I'm not really going into that detail. Now the last part is that if you keep the IDPs idle for a prolonged time, you do not provide enough partners, enough functions, so then what will happen? They will self-assemble. They will form insoluble aggregates, and these aggregates will deposit on ganglia or nerve glands, and it will lead to the fibril. This is the neurofibrillary tangle or plaques, as you all know, all familiar with Alzheimer's, Parkinson's, Huntington's, schizophrenia, all these neurodegenerative disorders. So the bottom line is you cannot keep for a cell, obviously I'm talking this to a cell, you cannot keep your IDPs idle for a long time, or a system, you know. And then if you just draw some analogy into the social context, actually there is a social entropy theory attributed to Dr. Bailey in 1990s. So one class of people, they tend to perform routine functions, so they are so-called the mainstream. Then there are other class of people, they do not have, for some or the other reason, I'm not really going into what is the reason, but they do not have any routine function to perform. They are kind of free, and they're idiosyncratic, they're highly individualistic, they have peculiarity unique to certain individuals, and they are the ones. Then if you suddenly encounter an accident, or you would see that these people are coming and helping people out, and but if they are not really having enough occupation or enough engagement, then what will happen? They will gather around, and then there will be some some social disorder. They will form some social disorder. So it's always a balance between order and disorder. That is important. Never a purely ordered society, may it be in molecule, in cell, may it be in the macroscopic society that of ours, never a completely disordered society, but it's an interplay between the two, and all should respect each other. It's not that the mainstream is better than the alternative, or the idiosyncratic people, or the other way around. And sometimes, this is my last slide, sometimes if proper attention has not been given, one key example I can tell you, this is of P53. P53, we all know, primarily is as a tumor suppressor, but it has many other functions as well. I mean, mostly related to growth arrest, DNA repair, etc. But it is seriously important for normal functionality of a cell in normal circumstances, where there is no adaptive stress that is in the system, or less, minimum, I mean manageable adaptive stress. But if the stress is too much, then it is one of the most deadliest proteins. The mutations are cancerous, and we all know that it's a very deadly protein. I'm sorry. It's a very deadly protein when not in a good mood. So similarly, we have seen in history that disorder can often lead to unique idiosyncrasies of a notorious and that of a genius. We can think of the life of Pablo Picasso, you can think of the life of Vincent van Gogh, several I can name, even Adolf Hitler. So, I mean, they're extremists in their own terms, but we can at least say that they were not really a commoner. They have several peculiar or idiosyncratic features that made them bit separate. So that's another feature of disorder, which is I found it was very astonishing as an analogy. So there I want to end. I hope I kind of could convey my idea to all of you, and I want to acknowledge accuracy, sir, although I can't see anyone here around. But obviously to our HOD, my dear friend Dr. Kuntal Kanti Goswami, and all the faculty members, and he's my friend. He's helping me. I had a lot of discussion with him about this. He has cracked Marie Curie fellowship recently. He was a former student here. And the illustration that which you see here, that was done by one of my relatives, Shubhangi Rai. So acknowledgement to all of them, and to my PhD supervisor who could open up the whole avenue of proteins in front of me, and I never found boring till now working with proteins. Thank you.
Okay, so thank you, Dr. Basu, for this excellent and engaging presentation. A very really excellent presentation, and really it is like a popular seminar, I can say, rightly. So now the session is open for questions. If any one of you have any question, you can ask. If there is any question, though we are running out of time. So, since Sai is always available with us,
I think we can.
So if there is any question, you may ask. Anyone?
Maybe later.
Okay. So if there is no question, then we can conclude.